goodness, you are never going to believe what our boss just told me. Our boss just said that if I don't get vaccinated, I'm going to lose my job. Apparently it's a requirement now. I don't even understand why it's so important. I have a whole bunch of problems with the vaccine. You know, first of all, it's so small, it's not even going to do anything. It's not going to do any good if I get vaccinated. The mRNA or whatever, it's just going to go away in my body. Even if it doesn't, what about the variants? Everybody's so worried about variants, so why wouldn't the vaccine cause the same problems? Okay, so say that I do get the vaccine, or maybe I don't. Maybe I get sick. It does the same thing. I don't see what the big problem is. I've had COVID, so I'm just as immune as anybody else is. And then say I do get the vaccine and it causes all these side effects. I want to get pregnant. That's a big deal. What if I can't have a kid anymore? And what if it causes all these health problems? Like I've heard of it causing problems with your heart. I don't know. I just don't think it's worth the risk. It's definitely not an option for me.
I think I can help here. I understand you're concerned and all about the vaccine, especially because it's new, which is a valid concern, but my associates and I will take a little time to explain, help you understand, and hopefully you'll be a little less hesitant. What's your first concern?
Well, first of all, I'm skeptical that the vaccine even does anything. I mean, I know it's a new vaccine and all, but how does a little bit of RNA or whatever even actually do anything in my body?
I'm glad you asked. This is my primary point of interest, so allow me to further explain.
The public perception of the COVID vaccine, and maybe other common vaccines for that matter, might include the instability or the weakness of the vaccine itself. The concern is that the mRNA and all the genetic material is going to immediately and completely degrade upon insertion into the muscle. Well, this is not completely true. Yes, when the vaccine gets injected into the muscle tissue, it does break down, but not to the extent that people may think. When vaccines are given, they need to be given in a site that allows for the correct bodily functions and organs to be involved. Vaccines are typically given in the upper arm. This is known as intramuscular injection. Muscles have immune cells and they have good blood flow, and this makes them useful because it essentially provides a direct route to the immune system. So, whatever antigens or immunogens we present to the immune system via intramuscular route, the body will begin creating antibodies to fight them. mRNA as a vaccine platform really began its trajectory in the 80s. The whole principle behind using mRNA as a vaccine platform mostly has to do with its nature as an intermediate messenger. We all know cells have genetic material including DNA and RNA. mRNA is one of the crucial RNAs that serves as a middle ground between DNA and proteins. It is one of the genetic materials that carries a code, and that code can create a protein, with the use of cellular machinery, of course. It is actually quite intuitive that you could use this coding principle to create target proteins, like say an antigen or immunogen from a vaccine. Yes, mRNA are generally thought to be unstable because of enzymes present in the blood plasma like RNases, which break down RNA, and because of immune system functionality when it senses a foreign object or substance. With decades of research, scientists have found a way to stabilize mRNA. There are 5 strategies used in the Moderna and Pfizer vaccines to make the mRNA more stable. The first 2 strategies target the 2 ends of the RNA strand, which is the 5' capping and an addition of the polyadenylation tail. RNA has 2 ends, one called the 5' end and the other called the 3' end. The 3' end has a series of adenine bases called the poly A tail, and the 5' end has a cap, which essentially keeps the RNA from being broken down. The 5' cap is a protein that does exactly what bottle caps do. It protects and keeps its contents safe. Think of these as some sort of barrier to prevent the important stuff in the middle from being accessed. If the tail of adenines is elongated, it becomes more stable and additionally promotes translation. Translation is the creation of proteins from the mRNA strands. A third strategy is called nucleotide methylation. Methylation involves the addition of a methyl group to the RNA. This CH3 is 3 hydrogens attached to a carbon. The presence of these methyl groups provides a signal that affects gene expression. These 3 strategies act as physical barriers to protect and regulate the transition of the mRNA. The 4th strategy is a method using optimal codons in the sequence. Codons are what 3 nucleotides code for in the creation of a single amino acid, and the accumulation of many amino acids creates a protein. These are essentially better codons that can be used when considering translation or the protection of the mRNA against degradative enzymes. Optimal codons avoid the untimely use of stop or start codons, or an example that both the Moderna and Pfizer vaccines have used is CGG as an optimal codon. CGG replaced most CGN codons. CGG is used more frequently, and there are more human tRNA that decode CGG than CGN. This is a part of the G-ending codons that are considered to be more optimal for tRNA abundance affecting translation properties. This is just one of the numerous codon optimization strategies and theories that are applied in the mRNA vaccines. Lastly, the COVID vaccine uses nanoparticle technology. These nanoparticles are comprised of lipids and encapsulates the mRNA. These are useful because it presents the antigen to the immune system much like a virus particle does. These are termed virus-like particles. These 5 strategies work together to keep the mRNA intact as it moves through the body so that it can adequately interact with the immune system and produce long-lasting immunity against the COVID-19 pathogen.
Okay, so I understand that the mRNA actually sticks around in our bodies, but doesn't that cause its own problems? I mean, if it's viral RNA, isn't it going to mutate and change in our bodies and create all these variants just like the virus does?
Yes, that's a valid concern if the vaccine were composed of an actual live virus. Even then, though, it's unlikely. Let's take a look at the vaccines.
But before we look at the different types of vaccines, let's back up a little bit. Why do variants even exist? Where do they come from? Upon infection, viruses and their components are copied over and over in order to make more viruses. As the genetic material is replicated, with every replication cycle, there's a chance for mistakes, known as mutations. When enough mutations accumulate and alter the way a virus functions, it becomes a variant. Think of the popular icebreaker game, telephone, where a word or phrase gets passed from person to person until, when it reaches the last person, it is distorted or altered. These types of mutations are random. They're not necessarily caused or triggered by anything, they simply happen, as mistakes often do. This is why viruses innately have the ability to mutate constantly and lead to variants. Knowing this, let's jump in and look at the different types of vaccines. We'll see why most are incapable of creating variants. There are 2 primary kinds of vaccines, whole virus vaccines and viral component vaccines. Let's start with the viral component vaccines. First, we have nucleic acid vaccines. These vaccines use genetic material, either RNA or DNA, to provide cells with the instructions to make an antigen. In the case of COVID-19, the vaccine contains a segment of RNA that encodes for a specific surface protein from the SARS-CoV-2 virus, known as a spike protein. The RNA is delivered within a lipid nanoparticle to the cell. Once the RNA gets into human cells, our cells make the spike protein, which is the antigen that triggers a specific immune response. This response builds immune memory so your body can fight off SARS-CoV-2 in the future. Some advantages of this type of vaccine are that they are easy to make and cost-effective. Also, since the antigen is produced inside our own cells and in large quantities, the expected immune response is strong. The main disadvantage is that these vaccines need to be kept at very cold temperatures of about negative 70 degrees Celsius. That's almost negative 100 degrees Fahrenheit. Approved COVID-19 vaccines of this type are Moderna and Pfizer. There are yet others that are still in clinical trials, which include CureVac and Inovio Pharmaceuticals. Next, we have protein subunit vaccines. This type of vaccine contains proteins from the SARS-CoV-2 virus, which are recognized by the immune system to trigger a response. The vaccine could contain protein fragments, whole proteins, or even many different protein molecules packed into nanoparticles, as is the case with virus-like particles. The advantages of this type of vaccine are that they have very good safety records and minimal side effects. However, the disadvantage is that the immune response is typically weaker, so they often need to be paired with enhancers, known as adjuvants, to help boost the immune response and ensure a lasting protection against the virus. Adjuvants can improve immune response, but this can also worsen side effects. COVID-19 vaccines of this type currently in clinical trials are Novavax and Sanofi. Viral vector vaccines are similar to nucleic acid vaccines in that they deliver genetic material to the cell that encodes for an antigen that will induce an immune response. The difference is the delivery method. As we discussed before, nucleic acid vaccines use a lipid nanoparticle. Viral vector vaccines use an unrelated virus to deliver the SARS-CoV-2 genetic material. Viral vector vaccines typically mimic natural infection and trigger a strong immune response. However, since there is a chance that many people may have already been exposed to the viruses being used as vectors, some may be immune to it, making the vaccines less effective. Vaccines that use this platform include Johnson & Johnson, AstraZeneca, CanSino, and Gamaleya. Of these, only the Johnson & Johnson vaccine has been approved in the U.S. After looking into how these viral component vaccines work, let's back up to the real question. Can any of these cause variants? The answer is no, and the explanation is simple. Each of these platforms use only fragments or components of the SARS-CoV-2 virus. These fragments are not viable on their own. They do not replicate as whole viruses and therefore cannot lead to variants. Now let's take a look at the whole virus vaccines. Here we have two kinds, inactivated and live attenuated. Inactivated vaccines contain killed SARS-CoV-2 virus particles whose genetic material has been destroyed. The killed virus is recognized by the immune system to trigger an immune response without causing illness. Because the virus is killed beforehand, it cannot replicate and therefore cannot cause variants. This type of vaccine is often recommended for people with compromised immune systems because they produce minimal to no side effects and are extremely safe. However, they often need to be delivered with adjuvants to boost immunity, since the immune response is not always strong enough to ensure protection from the virus. Vaccines of this type that have been approved elsewhere in the world include Sinovac, Sinopharm, and Bharat Biotech. Finally, we have live attenuated vaccines. These vaccines use a weakened form of the virus that can still replicate without causing illness. Because it is weakened, the pathogen will not spread and cause sickness, but the immune system will still learn to recognize its antigens and know how to fight it in the future. Since these vaccines introduce an actual live virus into the body, it is an excellent stimulant for the immune system that very closely resembles natural infection, without the downsides of actually having the sickness. Because of the strong immune response they cause, live attenuated vaccines can result in lifelong immunity with just 1 or 2 doses. This is longer than most other types of vaccines. While unlikely, the live attenuated vaccine virus does have the potential to revert to a form capable of causing disease. Mutations can occur when the vaccine virus replicates in the body and develops mutations that may result in a more virulent strain. Again, this is unlikely since the vaccine's virus's ability to replicate is limited. Even in its low likelihood, however, it is taken into consideration when developing an attenuated vaccine. Live attenuated vaccines are a well-known approach. Vaccines for measles, mumps, rubella, yellow fever, and some influenza vaccines are made using this platform. Rest assured that the development of these vaccines requires extensive testing before approval. Codagenics is an example of a live attenuated vaccine for COVID-19 that is currently in clinical trials but not yet approved. As you can see, there's a lot of different types of vaccines, but most can even cause variants. Even the type of vaccine that possibly could cause variants, the probability is very low, and that is why they undergo extensive testing. Luckily, you have options. You can pick the vaccine that is best for you and that you feel most comfortable receiving. After all, any vaccine is better than no vaccine.
Okay, so the mRNA sticks around and it doesn't cause variants. But so why should I get vaccinated? I don't understand why I should actually go get a shot instead of just getting sick. I'm not scared of COVID, so what if I get a little bit sick and have a little bit of a cold for a few days? I don't see why there's any difference really.
Well, that's not exactly true.
Infection is highly variable. You've probably heard all kinds of stories of people getting COVID. Sometimes people get so sick that they end up in the hospital, and other times they don't even know they have it at all. Vaccination has shown to be a lot more consistent, delivering predetermined and constant concentrations of antigenic material in each dose. These concentrations have been studied, so we know they are really effective at consistently producing immune responses. Additionally, recent studies on vaccine efficacy compare the natural immune response to that which the virus induces, specifically in relation to something called adaptive immunity. There are 2 key components of human immune systems: the innate immune system and the adaptive immune system. While innate immunity works on all pathogens, adaptive immunity creates memory. It is specific to each invader. The immune system can then be further broken down into cell-mediated immunity and humoral immunity. In order to have sufficient long-lasting immunity against a pathogen, the body requires activation of both cellular and humoral immunity. And recent studies have shown that adequate and long-lasting protection against COVID-19 infection may rely very heavily on the adaptive cell-mediated response to the infection. This could be, in part, due to the unknown lifespan of antibodies. Okay, that all makes sense, but I still don't understand how scientists actually know that the vaccine is producing this kind of response, or how can they compare the two? It doesn't really make sense.
Well, that's a great question.
See, our cell-mediated immune systems have cells called T cells. There are two main kinds, CD4 T helper cells and CD8 cytotoxic T cells. CD4 cells produce neutralizing antibodies, as well as stimulate immune memory cells. Neutralizing antibodies are the antibodies that actually stop the pathogen from infecting our cells. CD8 cells directly kill pathogens, and when they are activated, they produce antivirals, or cytokines, called interferon gamma and tumor necrosis factor, or TNF alpha. The antivirals can then be measured through a blood test to indicate if and how strongly our cell-mediated adaptive immunity is responding to an antigen. With vaccine development, it's ideal to create vaccines that induce neutralizing antibody production and cell-mediated responses. The vaccines that have been created thus far target the same parts of the virus that we respond to with natural infection. Researchers have begun looking into the immune response for vaccination in comparison to natural infection by analyzing the levels of cytokines produced by each. Tests conducted on people with naturally acquired immunity to SARS-CoV-2 show high levels of CD4 and CD8 T cells in around 80% of cases. The cells responded strongly to the spike and membrane proteins of the virus. The more severe cases showed higher levels of CD8 cells, whereas the more mild cases have more CD4 cells. The Moderna mRNA vaccine elicited a response biased towards the interferon gamma and TNF alpha cytokines, which indicates that it produces high levels of neutralizing antibodies and a strong cell-mediated response. In a study using the Pfizer vaccine, researchers found that the vaccine produced 1.9 to 4.6 times more neutralizing antibodies than a naturally acquired infection. It also caused an immune response biased towards CD4 and CD8 T cell responses. Additionally, as we previously talked about, the vaccine is only a piece of the virus. It can't mutate or replicate inside of you, and it is developed to induce a controlled immune response. However, the full virus can mutate and could potentially lead to worse versions of the virus or severe disease. Unfortunately, not a lot is known about the long-term immunity of the T helper cell activation against SARS-CoV-2. Both in naturally acquired infections and vaccination, the time frame for immunity isn't well determined. However, both produce adequate and equivalent immune responses for short-term studies. While it appears natural infection and vaccination can produce similar immune responses, natural infection can result in further spread of the virus, formation of new variants, or life-threatening illness. Even if you can fight it off without getting too sick, you could infect someone who can't. Vaccination is a way to provide immunity without the risks that come with natural infection. Okay, well, I'm starting to understand how all of this works, but I saw on one of my friend's Facebook posts that if I get the vaccine, it means that I can't get pregnant anymore.
Well, that didn't work for me. I got the vaccine and clearly I can still get pregnant.
Huh. Well, maybe it was just a fluke. Who knows? Maybe you got unlucky.
Okay. Well, my associate here can explain what the vaccine actually does and why it's not a good contraceptive.
Okay, so before I explain why the vaccine doesn't cause any fertility issues, let me first explain how the myth even started.
In November of 2020, Dr. Wolfgang Wodarg, a German physician, filed a petition to suspend the use of all COVID-19 vaccines due to the realization that the spike protein on the SARS-CoV-2 virus was similar to the structure of the syncytin-1 protein. This is a protein that helps the placenta attach to the uterus during pregnancy. A spike protein is a protein that protrudes from the envelope of some viruses. It looks like these red spikes on a virus. It allows for the virus to bind to receptors on the surface of host cells, which will then allow the virus's genetic code to invade the host cell. The theory was that the vaccine would attack the syncytin-1 and result in a pregnancy loss. However, the spike protein and syncytin-1 are very different, so there is no relationship between these two proteins, and the vaccine will not interact with it. So when you get the vaccine, it actually teaches your body to attack the spike proteins on the virus, which causes infection of COVID-19. In no way will the vaccine mistake the syncytin 1 as the spike protein, since they are structurally different. Even when looking at the amino acid sequences of the two proteins, there is no similarity between them. If you decide to get the vaccine while pregnant, it is unlikely the placenta or fetus will be harmed. When you get vaccinated, antibodies are produced in your body. These antibodies will recognize the foreign virus in your body. The antibody will attach to the antigen like a lock and key and neutralize it. These antibodies will continue to move through your body and will help to protect you from future exposures of the virus. When pregnant, these antibodies could possibly cross the placenta through vertical transmission, meaning the antibodies produced by the mother will cross through the placenta to the baby. But don't worry, this is a good thing. Antibodies produced by both the mother and the baby will ensure that they stay protected from the virus. Still not convinced? Well, data from a UK COVID-19 vaccine clinical trials provided more evidence that the vaccine does not cause infertility. These trials used the Pfizer, BioNTech, Moderna, and AstraZeneca vaccines. Within the trials, 18,860 participants received the Pfizer BioNTech vaccine. 15,181 received Moderna, and 5,807 received the AstraZeneca vaccine. Pregnant people were not included in the trials, and those who participated were asked to avoid getting pregnant. However, out of the 39,848 participants that were vaccinated, 29 of them had an accidental pregnancy. This additional evidence further emphasizes that the vaccine is not correlated with infertility.
So you really don't want to use the vaccine as a form of contraception?
Don't do it.
All this information is great and super helpful, but I'm still really concerned about some of the side effects. I listened to a podcast the other day, and they said that getting the vaccine can increase your risk of having a heart attack.
Yes, so what you're referring to is a condition known as myocarditis. This is characterized by chest pain that's caused by inflammation of the heart muscle.
It's easily treated by a medical provider, so with ample treatment, it's unlikely to cause severe damage to the heart.
But it can be caused by the vaccine?
No, so generally it's caused by a viral, bacterial, or a fungal infection, although there is a known association between the COVID vaccines and myocarditis.
I don't really understand. To be honest, you spent all this time explaining that the vaccine is just a piece of the virus. It's just mRNA. And so I don't really get how it can do the same thing as a viral infection.
That's a valid concern. My associate here will help further explain.
Let's dive into the structure of the spike protein. We know that the spike protein of SARS-CoV-2 is responsible for adhesion and entry into our cells, and a single virus contains many spike proteins. The S1 region contains the receptor binding domain, which is the part with affinity to our ACE2 receptors. Remember, the spike protein is what the mRNA vaccine tells our cells to produce. One peer-reviewed study explored if there were any amino acid sequences within the spike protein that match up with those responsible for myocarditis. Online databases, such as the Immune Epitope Database, provide researchers with previously discovered information to reference in future studies. This particular site contains information on many amino acid sequences, including myocarditis-associated antigens. Just so you know, the words epitope, amino acid sequence, and antigen all refer to proteins.
Those CD4 and CD8 cells you learned about earlier have T cell receptors, referred to as TCRs. Autoimmune responses can be triggered if the TCRs are unable to differentiate between self and non-self antigens. Various other cells in the immune system will process potential threats to our health and present these antigens on major histocompatibility complexes, or MHC, on their membranes. The TCR binds to the MHC, and this bond induces a number of downstream immune responses. This interaction is where the researchers decided to investigate. Using the IEDB, 35 associated antigens were compared to the mRNA-encoded spike protein within the vaccine. Over 2,500 computer-generated amino acid sequences were used in this comparison. Their conclusion states no significant similarities between the spike protein and known myocarditis-associated antigens. Since proteins are three-dimensional, there are many areas of the encoded spike sequence that are not exposed to the surrounding environment. When considering the interior of the protein, researchers identified 4 sequences with high similarity. However, given the structure of the vaccine-encoded spike protein, it is unlikely that these sequences are capable of binding to immune cells to induce a myocarditis event.
You're telling me that the spike protein doesn't actually bind to cells the same way that the myocarditis-associated antigens do?
That's correct. If the spike proteins are not exposed, it makes it difficult for them to bind to receptors on immune cells.
There have been reported cases of myocarditis among people who have received the vaccine. It's important to recognize the ratio of these reports, however. For example, a UK study took into consideration all reports of myocarditis, pericarditis, and chest pain from the vaccine adverse event reporting system. They concluded that just 64 people per million between the ages of 13 to 17 reported these types of side effects. Now that's a ratio of less than 1/100 of a percentage point.
Wait, what? They made such a big deal of it.
Exactly. So it's extremely small percentage, and ratios are very important to take into consideration.
Okay, so let's review. The mRNA is going to remain stable in your body. The vaccine is not capable of altering your DNA, nor will it cause variants. Getting sick will provide some level of immunity. However, the vaccine will provide that immunity while also minimizing the risks that come with infection. The vaccine will not affect your chances of getting pregnant. And while myocarditis is associated with the vaccines, the data tells us that these events are highly unlikely.
Well, I really appreciate you taking all this time to explain everything to me. But I think I'm just going to do my own research.